Um, things do get more and more complicated in this class because whatever it is that we'll talk about for this set of exam is built on what we talked about for the last set of exam, which is built on what we talked about for the first set of exam. So things get complicated because of that, not because of anything else. I'll mention this again and again and again. If you find that you don't understand this material, please do ask questions. Don't ask outlandish questions. I have to curb the questions in the other section because people started going off on a tangent. But ask appropriate questions. If you don't want to ask in class, I have office hours. If you don't want to ask me, you can ask either Lexi or Jerome during their office hours. I highly recommend that you do go to them because, like I said, information is getting progressively more difficult in terms of integration of information. Okay? If I talk too much, if I don't explain things well, definitely ask me so that I can explain things to you better. All right? I only know what I do unless people tell me that what I'm doing is not good enough. All right. So this section is, so far we looked at the genes, right? Gene, genome, and then we looked at the molecular aspect of biology. So now let's look at cell. Since this is a molecular cell biology course, we'll look at the cell. What are the components of a cell? We'll focus on the membrane. If you notice here, the biological membrane is really important. And then we'll look at the skeletal system. Then we'll look at what is called intracellular trafficking or vesicular transport, how materials transport. So we'll look at the cell and see how things come in and go out of the cell, and then once in the cell, how materials move around. And that involves the membrane, involves vesicles, so, and it involves the cytoskeleton. Okay? So for today, hopefully everything that I say today should be a review. I'll add a few things here and there, but at least I think it's a true review. All right? So cell, by definition, all cells are surrounded or enclosed in a plasma membrane, not just a cell membrane. So we'll have to distinguish. When you say a cell membrane, it's any membrane in the cell. A plasma membrane is specifically the membrane that encloses the whole cell, right? That's the outermost structure of a cell. Okay? And anything outside of that is the extracellular matrix. We've already talked about that. For instance, a cell wall is the extracellular matrix. Okay? Which you should distinguish a plasma membrane from, let's say, the nuclear membrane, mitochondrial membrane, lysosomal membrane, and membranes that encases or contain all the other organelles in a eukaryotic cell. So that's the gist of this right here. Eukaryotic cells, of course, have membrane-bound compartments. Remember, we said that prokaryotic cells do also have compartments. It just happens that that these compartments aren't membrane-bound, right? But eukaryotic cells have membrane-bound compartments. That's the difference. So just a reminder of what a prokaryotic cell looks like. Now, still has a plasma membrane. Okay, we'll talk about membrane in a lot more detail. We'll talk about all of these organelles. Okay, I'll remind you, as a plant cell, an animal cell. Just a reminder, if you don't know what any of these are, chances are you don't belong in this class. So we'll go into details when we talk about the function of each type of organelle. So we're going to look at the membrane first. Okay? The membrane is composed of three major components. All three of these components are a part of the membrane. So I don't know what you learned before. Chances are you learned that a membrane is composed of phospholipids and some proteins in it. But that's not all what a membrane is. Right? A membrane or membrane structure is composed of that of the amphipathic lipid bilayer, meaning mainly composed of phospholipids, but also have proteins as well as other lipids in it, right? It's amphipathic, meaning that it has both a hydrophobic and hydrophilic components. Right? So anything that is amphipathic has both a hydrophobic and hydrophilic components. Many amino acids are amphipathic. Some proteins, many proteins are amphipathic. Right? Again, amphipathic is anything that contains both hydrophobic and hydrophilic components. In addition to the normal, and this is normally what you learn what a membrane is, in addition to that, you actually, well, which also includes membrane-bound proteins as well as sugar. Right? A lot of sugar. And then a third major component is something called the cytosolic submembrane protein meshwork. So these are proteins, complexes of proteins, lots and lots and lots of proteins that are related to the membrane. They're mainly peripheral membrane proteins and not integral membrane proteins. Right? So they're associated with the membrane but not inserted into the membrane. And there's a whole meshwork, there's a whole layer of it. And it's only found in the cytosolic side, so we'll talk about that. So all of this is important for all membranes. Eukaryotic cells, anyway. We don't know much about prokaryotic cells, at least I don't. So basically, again, you learn that here's a phospholipid bilayer with the, this is not to scale, this inner hydrophobic region is a lot thicker. And then you have the hydrophilic heads, right, which are basically larger polar molecules. It could be, let's say, serine. Serine is a phosphatidylinositol, phosphatidylserine is a phospholipid, right? So the serine could be, which is an amino acid, could be the hydrophilic head, okay? And then, of course, we won't go into more details about the characteristic of the membrane, but suffice it to say a cell membrane is fluid-like, right? The fluid mosaic model where fluid-like, it's kind of like a little thicker than water, but it's still fluid. So anything that is found in it moves around within the membrane. Okay? Notice that it's a bilayer. That means that here's one layer of phospholipid, here's another layer of phospholipid. Okay? In terms of phospholipids, there are a number, about half a dozen or so different types of phospholipids. Like I mentioned here, here's the serine, ethanolamine, serine, choline. Those are the hydrophilic heads right here, right? And then the tails will be composed of 2 fatty acids. And then there are a couple of there's a sphingosine. They're related to phospholipid, but we lump them all in one. We just call everything phospholipid. And what you need to know about this slide, all that you need to know is that there are about 5 or so different types of phospholipids. You don't need to memorize the different types. Right? However, if I say phosphatidylcholine, you know that it's a phospholipid. There are more, by the way. There's phosphatidyl, at least one more, phosphatidylinositol. But there are a few different types of phospholipids, and that's important later on when we talk about the different types of membranes. Yes, sir?
Are membranes made up of different types of these phospholipids, or is it just like one type to make up a specific membrane?
Different types. So we'll talk about how different membranes are different, and we'll come back to this idea of having different phospholipids types. So we'll talk about that. In addition to phospholipids, there are molecules called glycolipids, for instance, which is a lipid, right? But has carbohydrate residues attached to the lipid, so it's a glycolipid. You can have pure cholesterol, sterols of cholesterol embedded in there. And again, if you remember from intro biology, cholesterol actually adds to the fluidity of the membrane. Okay? So the more cholesterol that you have in the membrane, the more fluid-like, the more material can flow around the membrane. So if you want the membrane to be more solid, then you remove cholesterol, for instance. Okay? And there are cases like that can happen. If you were to take a bunch of phospholipids, for instance, this is the structure that it's going to make. A phospholipid bilayer is a natural, spontaneous structure. Again, if you have enough phospholipid molecules, just phospholipid, in a beaker, I can guarantee you that eventually you will get this spherical structure. Not circular, but it's a sphere. I hope you understand that it's a sphere, right? And so you have the inside or the lumen, and then you have the outside. Okay? And materials, even without help, certain materials can move into and out of through the lipid bilayer or the cell membrane fairly easily, and we call that process diffusion. And certain molecules like oxygen, carbon dioxide, urea, water can move through freely. I'm not sure about freely, but without aid. Okay, and we'll talk about how all of that works. Okay. However, there are other molecules that the cell membrane would prevent other molecules from moving across the cell membrane unless there's help. Okay, so we say that the cell membrane is selectively permeable. Permeable means that things can move through, right? But they're selectively permeable. That means that certain things can't move through. Okay, so what are the materials that can move through, that can cross by diffusion, and what are the materials that cannot cross by diffusion? Materials that can cross by diffusions are, here are the characteristics. They are small. Small is really important. And they can be either polar or non-polar. Usually if it's polar, the molecule can afford to be a little bigger. For instance, cholesterol. Cholesterol can diffuse across a cell membrane. But if it's a polar molecule, it has to be pretty darn small. For instance, oxygen, carbon dioxide, water molecule, urea, okay, ethanol. These are very small, polar but small molecules. But then, again, if a molecule is nonpolar, like cholesterol, like fatty acid, then they can be a lot bigger. Cholesterol is pretty big. It's pretty huge, right? Which still can cross. However, if you have a really big molecule, I don't care whether it's polar or non-polar, you can't jam it through the cell membrane. There isn't any place for that. Okay? So there has to be a certain size. So those are the molecules that can diffuse across. How about molecules that cannot diffuse across? Absolutely never ever can an ion. Anything that has a charge, not just an ion, but anything that has a legitimate charge. Not just polar. Polar can mean that something it has a partial charge versus a full charge, right? So anything. So don't say just polar, because a polar molecule can still cross, like water, urea, right? They're all polar, but we're talking about a charge. Anything that has a charge, even an ion, even the very smallest entity, which is a proton, cannot diffuse across the cell membrane. Okay?
Okay.
And obviously, then anything that is polar and large cannot. For instance, sugar cannot. Proteins cannot. Okay? So whether or not sugar doesn't have an actual full charge, but since it's big and it's polar, then it can cross. Okay? So other components, let's see. Okay. So other components, and this leads to, again, the other types of ways to allow molecules to cross. And you probably have heard of transport proteins, so then there are proteins that help molecules to cross. But before we talk about transport protein, let's talk about how are proteins related to, what are the different types of membrane proteins, right? The different types, two major types of membrane proteins are integral membrane proteins and peripheral membrane proteins. Integral membrane proteins sometimes are called intrinsic membrane proteins, and peripheral are called extrinsic. I hate those terms of either integral or peripheral membrane proteins. Integral membrane proteins are proteins that actually are inserted into have regions, either membrane domain regions, or have a fatty acid that is attached to the protein, right? And these are the regions that get literally inserted into the membrane. Whereas a peripheral membrane protein is a protein that interacts with either a membrane protein or the membrane itself, like number 7 here. This is a peripheral membrane protein. Number 8 here, that's a peripheral membrane protein. Everything else is an integral membrane protein. In terms of integral membrane proteins, there are 2 types. One would be transmembrane proteins, where you find that the membrane actually, the protein actually has domain on both sides of the membrane, whereas you also have other proteins that either show up on one side or the other, okay? And they're called either endoproteins or ectoproteins, depending on the side. So we won't go into that much detail, but basically, integral membrane proteins would have regions that are integrated or intercalated or embedded into the membrane. Was there a question over there?
You answered it. I was just wondering if transmembrane proteins were somehow subdivided into whether or not they went all the way through the plasma membrane.
Yes. And yes. Okay. So I answered that good. Okay. And so then we talked about single pass versus multi-pass, right? So here's 1 pass, here are 3 passes. Like this is a channel. So this is a typical channel where these passes contain alpha helices, right? This right here is a beta pleated sheet that folds in like this. So it gives you a nice lumen inside, so materials can flow through. So if you hear about a sodium ion channel, potassium ion channel, a channel is a channel, okay? Versus a pump is something that doesn't have a lumen inside. And this could be a pump right here. Materials can still flow through, but then it has to open up, just like when we saw the sodium-potassium ion pump, right? They move this way. Okay? What are some of the function of these proteins? They transport, certainly, and we'll talk more about transport. I'll remind you more about transport. So transport of materials across the membrane. And some membrane proteins have enzymatic activities. They're actual enzymes. Some are involved in signaling. So we talked about receptors, right? So within the signaling systems, we have receptors. Yes, ma'am.
On the previous slide, it showed that they were all transmembrane.
This?
The one after that.
After this?
Yeah.
Right.
So with these 3, is it always transmembrane?
Not always. Transport proteins are always transmembrane proteins. Receptors are always transmembrane proteins, but the enzymes are not. So that's a good question. So enzymes are not. Don't worry too much at this point whether they're transmembrane or not. Later on, when we look at very specific activities, then you will have to know whether very specific ones are transmembrane or not. But in general, all membrane transport proteins are transmembrane, and all receptors are all membrane receptors are transmembrane proteins. Cell recognition. A lot of that has to do with the fact that the carbohydrate residue, and somebody I think answered that when I asked that question. In addition to other functions, the carbohydrate residues on glycoproteins act as a recognition, entity, right? A marker. And then let's say another protein from the other side recognizes that. So for cell recognition, Intercellular joining. So if you have adhesion, anchoring junctions, when we talk about tight junctions later on, when we talk about focal contacts, it's so that two cells can hang on to each other. And then membrane proteins can also help attachment to the extracellular matrix, right here. Okay. In terms of transport, go back to the first idea. The transport, we already talked about diffusion. Diffusion is when you have the small molecules, either non-polar or polar, but they have to be very small, can move through the lipid bilayer. The other type of diffusion is called facilitated diffusion, either by having a transport protein, but the transport protein can either be a channel, like I said, or a pump in this case, right? So in both these cases we call it diffusion. One is just diffusion, and the other one is facilitated. Facilitated means that a transport protein is required. So then, by definition, really, what's the difference? Why are these lumped into the same activities? Why? So what is a definitive characteristic of diffusion? Concentration gradient, right? Diffusion is whenever molecules move down a concentration gradient, regardless of whether or not there is a transport protein. Okay? So diffusion, just pure old diffusion, without a transport protein, still molecules can move down a concentration gradient. A facilitated diffusion is when you still have diffusion, which is molecules or ions move down the concentration gradient. However, a transport protein is required. Why is a transport protein is required even though you have a concentration gradient? Because if the molecule is charged, fully charged, or an ion, or if the molecule is too big. That's why, right? We already established that fact, okay? Versus active transport. Now, active transport is a little more difficult to explain in terms of, definitely there has to be a transport protein, right? And the transport is, that's the active part, is that the transport protein has to help aid in the transport. And more than likely, you learn that active transport is always allowing molecules to move up the concentration gradient, which is for the most part true, but not always. Right? So active transport is when molecules are moved across the membrane, or molecules or ions are moved across the membrane with the aid of a transport protein. Okay, so far so good. Whether or not, regardless of whether there's a concentration, regardless of whether the movement is up or down the concentration gradient or no concentration gradient. Usually it's up the concentration gradient or no concentration gradient, okay? But the difference between facilitated and diffusion and active transport is the fact that for active transport, energy expenditure, there is energy expenditure, so you have to put in some kind of energy. Okay? That's a major difference. Not whether or not it's up or down the concentration gradient. Although for facilitated diffusion, you absolutely have to have a concentration gradient, and these entities would always move down the concentration gradient. So I split hair a little bit there to give you a full idea of the definition of these terms. Are there any questions about what I just said? Did I just make it more complicated? No, okay, good. One person always represents the whole class for me, if you care to answer. So, all right. So, something that you probably didn't learn, and we'll come back to this idea multiple times. So it's not just whether or not you have lipids, phospholipids. It's not just whether you have proteins. It's the ratio of lipids and proteins. And like I said earlier, it's also how much cholesterol in there, for instance, right? So it's not just a phospholipid, it's other types of lipids, and then how much protein is in there, Okay? So in general, what you see is that the more activities that is required of the membrane, the higher the protein to lipid ratio.
Okay.
Right? The more activities that the membrane carry out, the higher the protein to lipid ratio. And lipid, when I say lipid, it could be both phospholipid, could be phospholipid, glycolipid, could be cholesterol, could be any type of lipids, right? But the idea is that proteins actually carry out the function. Okay? So if a membrane, again, is active, has a lot of functions, then there will be more proteins in the membrane. So for instance, if you could see, for instance, the outer and inner mitochondrial membrane, you see the protein to lipid ratio for inner mitochondrial membrane is a lot higher than for the outer mitochondrial membrane. That means that the inner mitochondrial membrane carries out a lot more function than the outer. So what kind of function that is carried out that is so high in the inner mitochondrial membrane?
The electron transport chain, right?
I'm sorry?
Isn't that the electron transport chain?
Yeah.
Yeah.
So cellular respiration,
Yeah.
right? Not just the electron transport, but also oxidative phosphorylation as well,
Okay.
right? So that is exactly it. So for instance, the membrane for a normal cell would also have a fairly high protein to lipid ratio because you have a lot of transport in and out. But then if you look at something like a myelin sheath, if you don't know what a myelin sheath is, I'll just explain it to you briefly so you know that we have, for neurons have very long axons, and information have to travel down the axon. But sometimes our axons are pretty long, literally a meter, so a whole yard. For me, it's not that long because I'm short, but for a tall person, it could be very long. So in order for this information to travel down the axon, it'll take forever. So in order to expedite that process, then there are these membranes, right, that wrap around the axon that is called the myelin sheath. And what the myelin sheath is because what it does is that it cuts down the number of activities, basically, right, in terms of protein. So we talk about sodium, potassium ion channels. By cutting down the number of activity, then the information can just skip over these myelin sheath, right, into what I call a node of Grandier. Okay, so therefore, the idea is that the myelin sheath would not have a lot of activity, and if it doesn't have a lot of activity, then that means that it doesn't have a lot of proteins, and that's why the protein to lipid ratio is low. Is that kind and sort of? Those of you who have had systemic phys and other classes probably had no problem with understanding that. Those of you who haven't, is that kind and sort of clear enough, at least for you to understand? Okay. And then in terms of proteins as well as lipids, there are glycoproteins and glycolipids. And like I said, the sugar, the sugar residues or carbohydrate residues are very important. Okay? And here's the deal. Now we're no longer going to talk about a membrane as a single unit. We're going to divide it into even more details. Right? So a membrane, a lipid bilayer, so the grayish part represents the lipid bilayer. Is one layer is the other layer, okay? Is divided into a cytosolic layer or leaflet versus a non-cytosolic layer or leaflet, because there are two layers, right? So then there's a cytosolic. And if one layer of the bilayer is cytosolic, then the other one is non-cytosolic. If one layer is non-cytosolic, then the other layer is cytosolic. So a cell membrane, a lipid bilayer, always has one cytosolic and one non-cytosolic layer or leaflet. So far, so good? Yes, sir.
Does it matter which layer is the cytosolic one?
Yes, it does matter a lot.
Will it always be like the inner one, or can it be?
Right, so we'll talk about, it does matter. Okay, it does. In fact, that's the next texting now, so it does matter. Cytosolic. Everybody know the difference between a cytosol and a cytoplasm, right? So the cytoplasm, by definition, is everything between the nuclear membrane and the plasma membrane. Whereas if you take the cytoplasm and you subtract out everything that is insoluble, including organelles, cytoskeleton, and big complexes of proteins, then you will get the fluid and water, basically, and anything that's soluble in the water. That is your cytosol. The cytosol is the fluid phase of the cytoplasm. Okay? That's where the cytosol comes in. So the cytosolic layer is the layer that interacts with the cytosol. And so therefore, the other layer is non-cytosolic. That's by default. So if you look in the cell, you look at a picture of the cell, and any layer that is interacting with the cytosol, then you just designate that as the cytosolic layer, and then the other one. And so, that comes the next must, right? These are musts. So the cytosolic layer must always interact with the cytosol. Therefore, another must is that the other layer is always a non-cytosolic layer. And the third must is that a non-cytosolic layer would never, ever, ever interact with the cytosol. And the reverse is true, is the cytosolic layer would never, ever, ever interact with something that is not a cytosol. Those are the musts. Right? Yes, ma'am. Leaflet or layer, the same thing. So this is a leaflet or a layer. Okay, so far so good?
Can you repeat that first point? The cytosol layer must...
The cytosolic layer must always interact with the cytosol. Okay, and the non-cytosolic layer always interact. You should never ever interact with the cytosol. Okay, and those are musts. All right. Next must, and I refer to this. When it comes to glycoproteins or glycolipids, the carbohydrate residues always found on the non-cytosolic side of the membrane. Always, always, always. Never the cytosolic side. Okay. So even if it's in a cell, then the carbohydrates has to point in the lumen of the ER, that is actually the non-cytosolic, right, side of the ER membrane. Okay?
What, can you repeat that one more time?
Yes, carbohydrates are always on the non-cytosolic side of the membrane. Which leads to the next must. These are all, there are absolutely no exceptions to any of this, which means that all the reddish line and yellowish stuff right here, all of that is this a layer of proteins called the cytosolic submembrane protein mesh. Okay? Cytosolic means that it's always on the cytosolic side. Submembrane because it's below the phospholipid bilayer, and it's a protein meshwork. That means that it's a layer of it. So it's a cytosolic submembrane protein meshwork is always found associated with the cytosolic leaflet or layer of the membrane. Okay. So when we talk about, later on, it's really, really important when we talk about vesicular transport that each membrane has a proper cytosolic submembrane protein meshwork. And then the cytosolic submembrane protein meshwork interacts with the cytoskeleton, which is also in the cytosol. Okay. And we'll talk a lot more about the protein meshwork later. Oh, right now. So memorize this. We'll memorize it for now. These are all the functions. Later on when we talk about vesicular transport and more about translation, right? We're not done with translation. More about translation, transcription, translation. We'll come back to these activities. Okay, we'll come back to all of this, and which are functions of the again the cytosolic submembrane protein network. So what are the principal functions of a cell membrane in general? It's not just the cell membrane is not just a selective permeable membrane. It has a lot of functions, and these are the functions right here. And again, we'll memorize this, so the last two slides are really important. You need to know all these things, but luckily we have a lot of time to look at each one of these functions in more detail. Okay? So compartmentalization, we already talked about that. Defense and keeping the integrity of the cell. So a cell could be of different shapes, for instance, and also being selectively permeable to materials. That means that not everything can enter the cell. And once the membrane is destroyed, the cell dies. So that means that the membrane is really, the integrity of the membrane cell, the plasma membrane is really important for a cell to live. We already talked about that. Regulation of this membrane is actually important in regulation of activities inside the cell as well as outside the cell. So inside, outside, and then responding to signals. So this is how cells talk, and not so. The idea is that receptors and proteins, even though they perform most of the functions, but specific cell membrane in terms of lipids, right, are also very important in all of these activities. So it's not all about proteins. It's also about phospholipids and lipids. So what we're going to do for the next about 10 or so slides is just very briefly, very quickly, I'm just going to rush through all these organelles and their functions, okay? Because I am going to assume that's in the prerequisites Bio 1 and Bio 2. You've heard of all these organelles before. None of this should be new. I'm just reminding you. Okay, so I'm going to rush through so that we have a lot more time to talk about the neat stuff in terms of cytoskeleton. Okay? So, so first of all, the nucleus and endoplasmic reticulum, you know that they are connected, right? In fact, the outer nuclear membrane is directly connected to the ER membrane. And so you would have, in the nucleus, you would have, that's a storage of DNA material, that's where transcription occurs, okay? And then the RNAs are delivered to the cytosol, and then translation occurs in the cytosol, but then polypeptides get pulled into the ER, so that's where integration of membrane lipid occurs. That's where insertion of the protein into the membrane occurs. Since lipid biosynthesis, meaning the synthesis of and somewhat the breakdown of lipids, occurs in the smooth ER. So in fact, in a cell, all lipids are synthesized in the smooth ER. And that is also where lipids are added to proteins to form a lipoprotein. Okay? And that is also where carbohydrate residues are added to lipids to form a glycolipid. Okay? So, and detoxification of dangerous materials. So if you look at something like the liver detoxifies a lot of material, and a lot of that detoxification occurs in the ER. It occurs in the lysosome, it occurs in the peroxisome, but also in the ER. And then, very important, calcium sequestration. We'll talk about calcium sequestration several more times as we talk about other processes, including signal transduction as well as muscle contraction. Right? So these are all important functions of the nucleus and the endoplasmic reticulum. Just to remind you that the nuclear membrane, there is a cytosolic. Here's one lipid bilayer. Here's another lipid bilayer. So therefore, this is the outer nuclear membrane, and this is the inner nuclear membrane, right? And so this has two layers. So the outside layer is a cytosolic layer. So therefore, this layer down here is the non-cytosolic layer, which means that the condition within the perinuclear space is what? Is non-cytosolic. That means that for the inner nuclear membrane, this right here is the non-cytosolic layer, and this right here in the lumen of the nucleus is the cytosolic condition. So in fact, the condition within the nucleus is the same as the condition in the cytosol for the most part. Whereas the condition inside of ER is the same as the condition on the outside in the extracellular matrix. Right? That's how you should think about this. Okay? And because the condition meaning mainly ion concentration, right? Salt concentration. Therefore, the inside the nucleus, the material inside the nucleus is fairly free to move across into the cytosol through something called the nuclear pore. Okay. Now the nuclear pore is not completely free for anything to cross. Water and certainly ions and small molecules can cross freely, but there are certain molecules that cannot. For instance, RNA, right? We talked about how mRNA requires a cap and as well as a poly-A tail to be allowed to move across. Okay, so a nuclear pore is more than just a transport protein. It's a humongous pore. Okay. Golgi apparatus. Golgi apparatus is involved in two very important things. So here we say that in the Golgi apparatus, these are the things that occur. So covalent modification, sulfation, glycosylation, adenylation, and phosphorylation a lot. But the main type of covalent modification that occurs is actually glycosylation. Okay? And when we talk about protein synthesis, when we talk about modification of a protein, in the Golgi we'll talk about mainly glycosylation, which is the addition and removal of carbohydrate residues. Also, the Golgi apparatus, which is composed of different flattened structure here called the cisterna. So you have a cisterna, and then you have plural would be cisternae. So cisterna, you add an e to the end, it's cisternae. And you would have the cis phase versus the trans phase. So here's the cis phase, here's the trans phase, and vesicle would move along all of these cisternae right here when we talk about vesicular transport. Okay, and you notice that even though we lump glycosylation into one activity, but actually there are multiple activities. Glycosylation is divided into many activities. The endosomal system is actually connected to the endomembrane system. It remind you the endomembrane system includes the nucleus, the ER, the Golgi apparatus, lysosome, and the plasma membrane, right? And these are basically organelles that contain membranes that can be exchanged. So membrane material can exchange among these organelles. And since the plasma membrane is a part of the endomembrane system, therefore anything that is taken in by the process of endosome, by the process of endocytosis, is also a part of the endomembrane system. Okay? So materials can be taken in if they're too big, or if they don't have a transport protein to cross the plasma membrane, then materials can be taken in by the process again of endocytosis. Phagocytosis is a type of endocytosis. Okay. Pinocytosis is a type of endocytosis. So therefore, endocytosis is when materials out here, the plasma membrane get pinched in to form a vesicle. So a phagosome is a type of a vesicle. Okay. So terminology. And then anything that is taken in, material, either proteins, lipid, food. So when you eat, right? When you eat a piece of steak, you eat candy, right? This is food. Food in what form? Food in the form of carbohydrates, proteins, nucleic acid, and lipids, right? Gets taken in, and then the food material has to be degraded. So most of them are degraded very roughly in the lysosome. Okay, some of them are actually delivered to the Golgi apparatus without being degraded in the lysosome. Okay, and then once in the lysosome, food materials are further digested in the peroxisome, for instance. Okay. What I'm gonna, let's see. So this is rough. We'll go into all these details later when we talk about the exocytotic pathway. But endocytosis basically, yeah, you know, the process of endocytosis and exocytosis are very similar. So what I'm going to do is hold off in talking about this. Wait until we talk about vesicular transport and exocytosis, and then come back and remind you that the same process occurs when material comes in. How's that? Okay. So we'll hold off on this for now. We're not going to talk very much about the mitochondria and the chloroplast at all. Okay. Every now and then I'll mention them, but not much at all. Mitochondria are found in all eukaryotic cells, and the chloroplast is a type of plastid. So if you ever hear the term plastid is a bigger term, and then there are 4 different types of plastids, and chloroplast is one. Okay. So mitochondria, chloroplast or plastids, and the nucleus are the only 3 organelles in a eukaryotic cell that contains 2 membranes, an outer and inner membrane. Okay, so you have an outer and inner mitochondrial membrane, an outer mitochondrial membrane, outer and inner chloroplast membrane, and outer inner nuclear membrane. Okay. And some of the important things about, beside this, some of the important things about chloroplast and mitochondria, in addition to the fact that the mitochondria, this is where a lot of ATPs are being produced. In the chloroplast, this is where photosynthesis occurs, right? But in addition to that, do realize that both of them can proliferate. So a cell can make more mitochondria, can make more chloroplast, independent of cell division. If you think about cell division, when in preparing to divide, for the cell to divide, to go from 1 parental cell to 2 daughter cells, all the other organelles will be split up, will double in amount and then split off into 2 daughter cells, right? And so lysosomes, ER, the nucleus even, right? All these organelles will only increase just before cell division so that you can have everything and then become daughter cells again. However, the chloroplast, the mitochondrion, and actually the peroxisome as well, a cell can increase the number, the amount or number of these organelles without having to carry out cell division. Okay, that's very important. It's very obvious that at least not the peroxisome, but at least the mitochondrion and the chloroplast is because of the idea of the endosymbiont theory, right? Where these things are thought to have been bacteria, eukaryotic, prokaryotic cell, 200 million years ago, or 100 million years ago, however long it was, right? And then they were endocytosed, but then they were destroyed, so they stay inside the eukaryotic cell, and the eukaryotic cell decided that, hey, they are useful for the cell, and these cells thought, oh, this is protection. So then that's how mitochondria and chloroplasts became where they ended up where they are. Yes, sir.
Can you have carbohydrates or phospholipids in between the membranes of the organelles if they're not even making it into the cell on the non-cytosolic layers
Of these,
of those or that particular envelope?
there's no need for carbohydrates in between, so there's no need. And again, looking at the functions of carbohydrates, it's for either absorb water or to keep the cell well hydrated or for recognition, and neither of those activities are that important in here. Right? That's a good question, but and that's the answer is that they're not needed, so therefore no carbohydrate residues in them. Okay, are we good? All right, next. Peroxisome. Peroxisome is really important. Your cells will die if you don't have a peroxisome. Your peroxisome works overtime if in cells like cancer cell, for example, cancerous, is because of the fact that there are a lot of ROSs, or the bigger idea is something called free radical. So free radicals are molecules that are very active. And they're very active or reactive because they have either been reduced or oxidized. Reduced means that an extra electron or electrons are added. Oxidized means that one or more electrons are removed from the molecule. In either case, the molecule or atom is no longer stable. They're very reactive. They shake just like me with lots of coffee, right? So if it were a free radical, which is also in oxygen, we call that a reactive oxygen species. So that's how I explain ROS. And in terms of free radicals, there are probably more reactive oxygen species as free radicals in your cell than any other types of free radicals. Okay, and so then if a free radical if something that became a free radical due to reduction, what happened is that inside the peroxisome, that free radical would be oxidized. So meaning that if an extra electron were added, then inside the peroxisome, the electron is removed. And the opposite is true. If something was oxidized to become a free radical, then that entity, that free radical, will have an electron added to stabilize it inside the peroxisome. Okay. So that's and this is where metabolism of hydrogen peroxide also occurs, even though it doesn't say here. Hydrogen peroxide is used, it stores electrons. So electrons can be either removed or added to hydrogen peroxide to carry out reduction or oxidation to then stabilize free radicals. That's a mouthful. That's a bunch of different processes all crammed into one, but they're all related. So far, so good. So let me repeat the whole flow. Okay, molecules, when they're reduced or oxidized, when you don't have a redox reaction actually working in parallel, when something is either only reduced or only oxidized, right? Then it becomes a free radical. Then these free radicals will be taken into the peroxisome, and then either will be reduced or oxidized to get these molecules to be in a stable condition. And reduction requires an electron. Oxidation requires the removal of an electron. And in either case, then hydrogen peroxide metabolism has to be activated concurrently with stabilizing these free radicals. Okay. So therefore, we say that hydrogen peroxide is metabolized. Metabolized means either built up or broken down. Okay? A process of beta oxidation is important because this is where oligomers, meaning like oligopeptide, oligonucleotides, peptide nucleotides, carbohydrate, lipid, and sugar, oligosaccharides, right? So in the lysosome, complex carbohydrates, protein, nucleic acids are digested, but only in bite sizes. So, but then in the peroxisome is where these all oligomer are degraded into the subunits, the monomer. Right? So you might have an oligomer of, let's say, 5 amino acids long, and then in the peroxisome, it was cut down to 5 amino acids long in the lysosome, delivered to the peroxisome, and then that oligomer would be further digested or cleaved into individual amino acid. And the cleavage of the breakdown of these oligomer is through the process of beta oxidation. Again, that's a lot of explanation. Biochemistry major, chemistry major in here, you probably already know all of that, right? So, but for a lot of biology major, you probably haven't learned that. And then lipid synthesis as well, okay? Occurs in the peroxisome. Vacuoles are only really found in plants, as well as eukaryotic cells that have a cell wall like fungi. Right? Vacuole is the equivalent or similar to vesicles that we have in ours in animal cells. What the vacuoles do is they have a lot of, there are different types of vacuoles. There's food vacuole versus a central vacuole. Okay? So let's just focus on the central vacuole. Central vacuole is what allows us, when you see a plant stand erect or wilt, that is the activity of the central vacuole. Is that if there's enough water in the environment, the plant takes up the water and then place it into the central vacuole puffs up, and that's why the plant stands erect and is alive. And it wilts because you don't have enough water, so not enough water goes in, and so there won't be enough water to keep the plant cell taut, right? So then the plant will wilt. That's about it. So regulation of osmotic regulation, basically. Okay. So again, like I said, there are a couple of other types of vacuoles. For instance, a food vacuole. Food vacuole is where food material, the plants take up food and then it stores food in a vacuole to be used later. So far, so good. I'm going to continue, by the way. But are we good with all the plant parts? I mean, the cell parts. Do I need to slow down on any of that? No. You remember everything? No. Okay. We're about to talk about the cytoskeleton. You understand? A lot of time talking about the cytoskeleton. Okay. Cytoskeleton has a lot of functions. Just to remind you that we've talked about the cytoskeleton in a way before. These are entities that have individual proteins, they don't monomers, they don't do anything, right? But by the process of polymerization or assembly, polymer assembly, and disassembly, this is when cytoskeletons have function. So again, as monomers, they don't have any function. Well, most of them don't have functions, a few of them do, but they really have functions as a polymer. Okay? And there are 3 major types of cytoskeleton, 2 of which are here, and this is just a pretty picture to show you. Actually, yeah, 3 of them, 2 of them are here. So either an actin microfilament, that's a little redundant. Most of the time I just call it a microfilament. Okay, you've heard of these as actin filaments. So they're the same thing. Actin filament, actin microfilament, or microfilaments, the same thing. You've heard about them in terms of contraction. And then there are microtubules. Tubulin microtubules are just microtubules. Okay, so I call these things microfilaments and microtubules. And if you look at this picture, this pretty picture of fluorescence using fluorescent dye, you see that there are these green things that radiate outward from, here's the nucleus, That's a stain called DAPI stain, which stains for something, for anything that is acidic, and certainly the nucleus contains DNA which is acidic. So would you think that the DAPI stain would stain for mitochondria? Yes, because mitochondria have DNA. How about chloroplasts? Yes, you just don't see them here just because they're really small. Right? So you see the green materials that radiate out, right? That's the microtubules. That's how they are arranged in the cell. Whereas you see the reddish, yellowish, somewhat orangey material, they're on the and then they don't radiate out, right? They're more lined up this way, or on the fringe here they do radiate out a little bit, right? That's a microfilament. We say that this layer of the cell, so here's the outer, is the cell plasma membrane right here, okay? And I can tell, you can tell from here that this cell is a cell not in your body but in tissue culture, because here's the plate down here, and then the cell settles down, and that's where a lot of microfilaments are found, okay? And we'll identify that these are stress fibers later on. But you notice that the microfilaments are only found on the outer edge of the cell and not in the middle. So we call this area of the cell cortex. The cortex of anything is the outer edge, right? The cortex of a brain, the cortex of a spinal cord. If you look at, anybody who is or has taken histology, you know that other cells have, other organelles have, or other organs have cortexes, right? Your tonsil have cortex, your all of the that are called lymphoid organs and lymphoid tissues have cortex. Okay, so that's the cell cortex right here, meaning the outer edge of the cell, right? And really, microfilaments are only found there. Okay. So here are the three types of the cytoskeleton. You have microfilament, which is composed of small subunits called actin or G-actin. G is a capital letter G. G stands for globulin, meaning, you remember, proteins can be found in either a globular structure or a fibrous structure. So the G of G-actin stands for globulin. Microtubules are composed of tubulin monomer. There are actually different, there's a whole family of tubulins, alpha, beta, and gamma. So remind you, hopefully it's a good memory, remind you about gene families, that the alpha, beta, and gamma tubulin probably came from one gene that experienced gene duplication a couple of times, and then independent DNA mutation down the line through evolution, so that now you have three different tubulin members of the tubulin family that have similar functions, right, which have to do with microtubules. And then you have the intermediate filament, which there isn't a set monomer, because there are about half a dozen or so, 6, 7, 8 or so different subunits depending on cell type. So the subunits for intermediate filaments are cell specific. Right? Also, for the microfilament and microtubules, these are cytoskeletons that are only found in the cytoplasm. And here I use the term cytoplasm instead of cytosol, right? Because they're insoluble. Whereas intermediate filaments has both can be a part of both the cytoplasm as well as the nucleoplasm. Nucleoplasm is the area inside the nuclear membrane, right within. So intermediate filaments can be in both the cytoplasm as well as the nucleoplasm or the nucleus. To distinguish between the two, when intermediate filaments are referring to intermediate filaments that are found in the nucleus, I will call them the nuclear scaffold. Okay? in addition to having monomers being used to form a polymer or for polymer assembly, there are other proteins that are not, there are other proteins that are not G-actin or tubulin, right, that are associated with the cytoskeleton. Okay, so there are many cytoskeleton associated proteins. One of which is really important, is a family of proteins called molecular motors or motor proteins. Okay, these are proteins that help in transport of materials as well as actually help the cell move. Okay? Motor proteins or molecular motors are only associated with either G-actin, a microfilament or microtubules, and not intermediate filaments. So that would lead you to think that intermediate filaments are not involved in movement of materials in any way within the cell. Right? So when we talk about vesicular transport, for instance, definitely either microtubules or microfilaments are involved, and they definitely involve molecular motors. Let's see. Okay. So the next, so I'm going to do this in layers. Okay. You know what? I think we're on time. So let's stop lecture, and I'll just see you on Thursday.